This is part one of the 2 lectures you need on the muscular system. This is a very short section of the course. We cover everything in 2 lectures, and I focus the 3rd lecture exam solely on the muscular system. So it comes fast and furious. You have 2 quizzes, 1 lab manual assignment, 2 lectures, and then you have November 11 and 12 to take this lecture test. So the muscular system quiz you'll be able to take at the end of this lecture. The muscle tissue quiz, you'll need both this lecture and lecture 2 in order to take that quiz. Don't forget, all lab manual assignments are due by 5 p.m. on the date they're due, so plan ahead. We're at a place where I've been telling everybody this for 7 weeks, and we need to get on it. So here we go, diving into the beginning of our muscular system. So first up, as we start the muscular system, anytime you see prefixes or affixes of any sort with MYS or MYO, it's referring to the muscular system. We have 600 plus muscles in the body. 40% of our body weight is muscles, and muscles are considered organs, and that's an important piece. There's some golden rules here of skeletal muscle activities. All of your muscles are going to cross at least one joint, and the bulk of the muscle is going to lie proximal to the joint that's crossed. All of your muscles have at least 2 attachments. Some have many. We'll talk about that later. An origin and an insertion. And big thing to remember is muscles are pulling on the skeleton. Even though you might be pushing a door or pushing a person, right? Your muscle is pulling on the skeleton in order to create that action. And during contraction, the insertion is going to move toward the origin. And briefly, a review of our types of muscle tissue. We did this back in lab, it seems like forever ago now. We have 3 types of muscle tissue: skeletal, cardiac, and smooth. I'm going to go through each one at a time, help you fill in these boxes on this slide here. So first up, our skeletal muscle. The location here is the muscle tissue that is covering the actual skeleton. We have long fibers of all muscle types that are striated. Typically, we think of them as muscle fibers. They are voluntary and work with the nervous system. They have fast contraction, remembering that they tire easily. So oftentimes, we train our muscles to be able to sustain fast contraction for things like exercise. And they really are for movement and power, which is not necessarily rhythmic, but more about movement and power and being able to use as needed. When we think of cardiac, we're thinking of the heart. So our cardiac muscle tissue is located in the heart. It is also striated like skeletal, but it has the branching chains of cells, similar to what we saw when we did the labs and we looked at them 3 types of muscle tissue. It's involuntary, which is brilliant because otherwise you'd have to remember to say beat, beat, beat every time you wanted your heart to beat. So involuntary control, slow speed of contraction, and it is totally rhythmic. Again, thinking about that lub dub or the action of the heart and the pumping mechanism that occurs. And then finally, we have our smooth muscle tissue, which is located in the walls of organs and our blood vessels. Like your skeletal, they are long fibers, but it is not striated. They are also involuntary. And then we have our slow and sustained contraction. Some rhythmic, some are not. 4 key characteristics that allow muscles to perform their duties. First up, we have excitability or responsiveness. This is the ability to receive and respond to stimuli by changing membrane potential. And we'll get into that when we actually talk about the actual contraction, when we get into how a muscle actually contracts and the regulation of that, like so sliding filament theory of muscle contraction. So we'll talk more about excitability and responsiveness when we get there. Contractility is the ability to shorten forcibly when stimulated. Again, shorten forcibly when stimulated. Contractibility is that ability to extend or stretch. So we have shortening when contracting, but being stretched even beyond resting length when relaxed. So contractility and extensibility work a little bit hand in hand. And then elasticity, which is the ability to recoil and resume resting length after stretching. Okay? So the ability to recoil and resume resting length after stretching. 4 important functions in the muscular system. First up is to produce movement. What that means is for skeletal muscle tissue, locomotion and manipulation of the environment. For cardiac, it's sending blood coursing through our bodies. And for our smooth muscle tissue, that's digestion and urinary-based movement. We work to maintain posture and position. So your skeletal muscle is functioning almost continuously in order to counteract the pull of gravity and stabilizing joints. So the muscle is going to pull on bones to cause movement while strengthening and stabilizing those joints. And then for heat production, we generate heat as muscle contracts. Therefore, this producing heat piece helps to maintain our normal body temperature. So on the next slide after this one, you're going to see a nice diagram of the gross anatomy of skeletal muscle, and it's going to show you how it breaks down into these smaller components, but from a gross anatomy standpoint. Each muscle is an organ with several kinds of tissues, right? Your skeletal fibers are primary, but you've got blood vessels, nerves, and connective tissue also present. So with the connective tissue, we're talking about binding, protecting, strengthening. We typically see the combination of fibers to increase contraction strength, and it's often continuous with tendons that attach muscle to bone. And then we also can hold our blood vessels and nerves here. And we see this rule of threes, and I kind of talked about this at the beginning of the year, that we're going to see a lot of rule of threes in the layers of things that we study. So here's where it starts with the connective tissue sheaths, and you'll be able to see these 3 linings on the next diagram. So we have our epimysium, which is the outer covering the muscle, and notice you've got the MYS in all 3 of these, so we know we're talking about muscle sheaths as opposed to some other type of tissue sheath. So the epimysium is outer covering of that whole muscle, typically blends into the tendon or the fascia, which binds the muscle to the skin or the subcutaneous tissue. Let me say that again. That epimysium, made of dense irregular tissue, is going to blend into the tendon or a fascia, fascia, which binds your muscle to skin or subcutaneous tissue. The perimysium is kind of our middle layer, is going to bind groups of muscle fibers together into fascicles. The word fascicle meaning group of muscle fibers. It's also dense irregular tissue. And then the endomysium is going to cover your individual muscle fibers. It's going to cover each individual fiber made of areolar connective tissue and tend to be continuous with one another. So on the next slide, you can take a look at that diagram, and I've relabeled with some of the definitions for you so you can see that. Here's your diagram. You can see that tendon that's going to attach the muscle to the bone. You can see the muscle belly. So then it goes from your epimysium, which is covering that whole area, and then it gives you one fascicle or one bundle of fibers. That bundle is covered by the perimysium, so outer epi, peri is that middle, and then you see that broken down into a single muscle fiber, which is covered by the endomysium. Okay? And this is multinucleate, okay? And we're going to get into that smaller anatomy as well, the microscopic anatomy. So you'll see that it takes this single muscle fiber, shows you the sarcoplasm, which I'll define for you in a minute, and a myofibril, which we will talk about when we get to the microscopic anatomy. The diagram that we just looked at, right? We talked about the golden rules at the beginning. The origin and the insertion are going to determine the action, right? So the origin is dealing with the immovable or less movable bone. In your limbs, the origin typically lies proximal, closer to the insertion. The insertion is your movable bone, right? Again, so the origin is typically going to lie proximal in relation to the insertion. And then either the origin or insertion can be directly or indirectly attached. So what does that mean? Well, you're talking about a direct fleshy attachment. So the epimysium is fused to the periosteum of bone or the perichondrium of cartilage. That would be a direct attachment, directly fused. In an indirect attachment, the tissue wrappings extend beyond the muscle as a rope, like a tendon, or a sheet, like an aponeurosis. This is more common due to durability and size. So indirect attachments are much more common due to durability and size. Functional groups, and this is how muscles can act in one of three ways. And if they're acting like number one in one type of movement, they may act as number two in a different type of movement. Let me explain. Your prime mover is the muscle that is producing a given movement. It's the leader, right? And you can think of this like your English class, right? You got your hero here. And you have the antagonist. So you have your opposing muscle to the prime mover, the one that's against the leader. Okay, so when the prime mover is active, the antagonist is going to stretch or relax. Okay? Then we have a synergist, which is a group of muscles working together to perform a movement. So they're going to help the prime mover by adding force or assisting the given movement, or even eliminating an undesirable movement. Right? So a synergist is going to help, the helper. So it's either going to assist in continuing the same movement or eliminate undesirable movements while that prime mover is contracting. Okay? Your fixator is a synergist that specifically immobilizes a bone. So the stabilization of a joint allows the prime mover to function. That would be a fixation. Then on the bottom of this slide, you can see I give an example of your bicep muscle. It's the prime mover of elbow flexion, the antagonist of extension, and then the tricep, prime mover of extension, antagonist of flexion. So you can see where these two work in pairs, create desired movement. All right, so how do muscles get their names? Well, there's a large number of criteria, right? And so you're going to get to a place where you're going to have to learn a lot of these, and there's a lot of videos posted already in the module for lab side of things, but I'll give you a couple recommendations after I tell you a little bit about this. So the muscles are named for a number of criteria. Okay? First are locations, so the bone or body region associated with the muscle. For example, the temporalis overlies the temporal bone. Pretty good. Tells you the name of the muscle, tells you where it is. Okay? The second is by shape. Oftentimes the muscles have a distinctive shape. Right? So the deltoid equals triangle, and the deltoid is triangular in shape. So that's also a nice way of thinking about that. The relative size of the muscle, or the size in comparison to other related muscles. So maximus, largest, minimus, smallest, longest, long, brevis, short, right? So those are four words you could see. So you think of that as the gluteus maximus versus the gluteus minimus. So we have the relative size there. Our fiber orientation has to do with the orientation of the muscle fiber in relation to the body or bone axis. So rectus, being straight, means it runs parallel, and versus means it's going to run at a right angle. That helps. We could have the number of origins. So the heads are attached to different origins for the same muscle. Bi, 2, tri, 3, quad, 4 tells you how many origins that you're going to have. And the location of the attachment or the points of the origin insertion. Origin is always going to be named first. And then finally, it could be named for what it does or the movement that's produced. So how are you going to learn all of these? Okay, so first you've got to pay attention to what the name actually is. Be alert to information provided by the actual name, like we talked about with temporalis overlying the temporal bone. Read the description and identify the muscle on the figure, right? Help look at the visual, figure out what it looks like, and see if that helps you remember what it's called. Relate that muscle's attachment and location to its action. This is going to give you some functional details that might help you remember where it is and what it's called. And then keep in mind, act out the movements yourself while feeling for the muscles contracting beneath your skin. The book has a large number of tables, and I believe you've got your handout for lab and all the videos that are posted. So as you look through the book, there's pages upon pages of muscles, descriptions, origin, insertion, and action, and it even includes the nerve supply. So make sure you take some time to take a look at all of that and watch all of the posted videos. So I mentioned on the previous slide on muscle naming that we were dealing with fiber orientation, and now we're talking about how we have an arrangement of the bundles of fibers to determine the range of motion and the power, right? So your bundles of fibers determine range of motion and power. If they're longer, they're more nearly parallel, and the more they can therefore shorten, right? So we have 4 very common arrangements, and 2 of those have some supporting or more detailed explanations as well. So first up, let's do circular. These are ones that are going to be arranged in concentric rings and typically surround external body openings. They close by contracting, things like sphincters. And then we've got 2 orbicularis oris or orbicularis oculi. Those are surrounding the mouth and the eye. Our convergent are broad origin fascicles that converge toward a single tendon of insertion. They're either triangle or fan shaped. So your pectoralis major would be an example of a convergent arrangement. We have a parallel. So the length of the fascicles run parallel to the long axis of the muscle. The muscles are strap-like, like the sartorius, or spindle-shaped with an expanded belly, like a biceps brachii. Your spindle-shaped are your fusiform. So where it's got fusiform listed there, you're talking about spindle-shaped parallel arrangements. So they have that expanded belly. Something like your biceps brachii would specifically be fusiform. Your pennate are your short fascicles attaching obliquely to a central tendon running the length of the muscle. They come in 3 forms. Uni, so the fascicles insert into one side of a tendon, like your extensor digitorum longus. Bipennate, insert into a tendon forming opposite sides, so it looks like a feather, like your rectus femoris. And your multi, which looks like many feathers side by side with quills inserted into one large tendon. It would be like your deltoid. There are 2 main categories of contractions, isotonic and isometric, and they depend on whether a muscle changes length or not. Your muscles are going to change in length in an isotonic contraction, but they are not going to shorten when you do isometric. So your isotonic contractions result in the change in muscle length. You have 2 different kinds here. You have concentric, where the muscle contracts and it shortens, and you have eccentric, where the muscle contracts but it's lengthening. So your concentric is a basic picking up a book, kicking a ball. Eccentric is really going to be used for coordination and purposeful movements. The muscle here that's contracting but is lengthening, think of when how your anterior thigh works as you're walking down a steep hill. The actions are actually 50% more forceful, and they're actually more often to blame for the delayed onset muscle soreness. So if you think about being sore after you go hiking, typically that's going to be the downhill return, not necessarily the uphill climb. Really great example depicted here in the picture is the use of bicep curls, where you get concentric and eccentric working together. So you get a concentric contraction in the biceps brachii during the upward phase, and then you get an eccentric contraction during the downward phase. It also gives you an example of how that could be working in an isometric contraction when you're just holding that weight still in a partially contracted phase. In your isometric contraction, right, so we're not talking about the change in muscle length. So you're really not having any movement occur. Right? Muscle tension is going to develop, but the load isn't moved because the load is greater than the force. Or in the case of that picture, that you're not moving, you're just holding that weight still. Typically, you see isometric contractions holding your upright posture, keeping other joints stable during movement, and those types of things. But again, you can create an isometric contraction. You also could do a wall push, where you push against the wall. You're not going to move the whole wall. You're creating an isometric contraction. So lever systems here, we're dealing with a little bit of physics, and I was trying to find a nice video, but it didn't really work out in my favor. I didn't like any of the ones that I saw, but you can do some Googling for a little bit more information on this one. That would be fine. But here's our basic definition. We have 4 parts to our lever system of movement: lever, fulcrum, effort, and load. So it's a rigid bar, known as a lever, moving on a fixed point, your fulcrum, when force, which is the effort, is applied to move a resistance or a load. So a lever allows the given effort to either move a heavier load or move that load farther and faster. And that's the difference between a power versus a speed lever. So your power lever is going to have the load close to the fulcrum and the effort applied far from the fulcrum, with a small effort moving a large load. Power levers tend to operate at mechanical advantage. You can see that as a class 1 lever. Your speed levers, your effort is applied close to the fulcrum, or closer to the fulcrum than the load being moved, and greater effort is required. So you have a larger effort to move a small load, but the load arm is longer than the effort arm, so you get greater distance at greater speed. This works at a mechanical disadvantage. So we have our 3 classes of levers here, and I've shown you examples both kind of in your everyday life on the left and in the human body on the right. For your first class lever, you have the effort applied at one end and the load at the other, with the fulcrum somewhere in between. So this is your example of like a seesaw or your scissors, or in the body, lifting your head off your chest, that head neck thing. Second class lever, you have the effort applied at one end and the fulcrum at the other, with the load in between. These are your wheelbarrows. It's more uncommon in the human body, but it's the kind of the action of, from a standing position, going up on your toes. These are levers of strength. Your second class lever is going to be really about strength. And a third class lever, you've got your effort applied between the load and the fulcrum. And these are speed levers, and they're always at a mechanical disadvantage. And here you're seeing tweezers or forceps, or in the picture, your shovel. And most of your skeletal muscle is going to be a third class lever. They're typically thicker and more powerful, but you're thinking about speed. Yeah. I'm going to do part of our meat and potatoes here, kind of the big piece of this as we head towards sliding filament theory of contraction. But I'm going to just start with our microscopic anatomy and getting some of the definitions and understanding of the muscle fiber out of the way first. End our lecture, our first lecture there, and come back and focus in on the sliding filament theory next time I post for you. So your muscle fiber each fiber is a long, cylindrical, multinucleated cell containing calcium-regulated molecular motors just beneath the sarcolemma. And our sarcolemma is our cell membrane around the fiber. I've posted that definition right here on this slide because I know it can be troubling a little bit. So it has the same organelles as other cells with 3 specialized organelles, which I'll talk about on the next slide. Cells here have an increased length and diameter, so they're 30 centimeters long, and that's huge because we have hundreds of embryonic cells that fuse together to produce each fiber. And they're 10 times the size of an average body cell, kind of big. And here we're talking about them being striated, which we've talked about a bunch. I already mentioned sarcolemma, that's your cell membrane, and your sarcoplasm, which is similar to cytoplasm, but with more glycosomes and myoglobin. Your glycosomes provide glucose during muscle cell activity for ATP production. Okay, one more time. Glycosomes provide glucose during muscle cell activity for ATP production. And your myoglobin is your red pigment used for storing O2. 3 specialized organelles. We have first up our sarcoplasmic reticulum. This is an elaborate form of your ER. Remember, part of a cell, endoplasmic reticulum. So your sarcoplasmic reticulum is a specialized part for muscle fibers. It's a membrane channel that runs longitudinally in the fiber for storing and regulating calcium release for contraction. And when we get to our next lecture, you'll see where that plays a big role in how it is calcium regulated. The T-tubules are channels that run transversely to the sarcoplasmic reticulum, and they're going to conduct nerve impulse deep within the muscle. It's really there to increase surface area. And then the big one, which are myofibrils. They run the entire length of the fiber. There could be hundreds or more in one muscle fiber, and it makes up 80% of the cellular volume. And this is where we get the ability for the muscle to shorten. So we are made up here of chains of sarcomeres linked end to end with smaller myofilaments. The myofilaments are a protein. And then you have striations and our actin and myosin. So striations, so we have these repeating dark and light bands that are evident along the length of the myofibril. Your dark bands are called A bands. And there's a lighter part within it called an H zone bisected by the dark M line. Now again, remember, hold on, pause. You're going to get to see this on the diagram on the next slide. So let me say that again. Striation. We have repeating dark and light bands throughout the length of this myofibril. Your dark portions are A bands, and there's an H zone bisected by a dark M line. Your light bands are called I bands, and they have a dark midline known as a Z disc. Between that, the Z discs is the sarcomere, the smallest contractile unit of muscle fiber. And then you have your thin and thick bands. Your actin is thin, containing the microfilament. Myosin is thick, which is motor protein. And the myosin extends the entire length of that A band. The two of those together, actin and myosin, play a role in motility and shape change in almost all cells in the body. So let's take a look at the microscopic anatomy on the next slide and try to clarify what all of that just was. It can be very confusing. Here we are, looking at this diagram, this microscopic anatomy. So if you think back to the first picture, we had the sarcolemma, right? So the cell membrane around one single fiber, part of that muscle fiber. And we are taking a look at what's called the myofibril, right? So this is our contractile unit. The myofibril is what's responsible for contraction, right? So there's these chains of tiny contractile sarcomeres. Okay, so we were talking about striations. You can now kind of see how these striations work. So that A band there is the dark band with a lighter section known as the H zone and the dark M line. That's your A band. You can see that it's surrounded on either side there by the I band. These are your dark sections, dark bands, with the Z disc in the midline, which is lighter. And that Z disc is your legitimate sarcomere, that smallest contractile unit. Okay, you've got your T tubules listed on here, and your sarcoplasmic reticulum is listed on here. So your T tubules that run separate, kind of that overlay section there you see that it's blue. And then, or in yellow. The yellow is your T tubule, sorry, your blue is your sarcoplasmic reticulum. So you notice that your T tubule is running one direction and and it's just a band, and then your sarcoplasmic reticulum is running in the opposite direction. Okay, it's got some other things listed on there. You should know what mitochondria are, right? Powerhouse of the cell. And it's got what's called a triad. That's just a meeting section between your T tubules and your sarcoplasmic. Okay. So this is kind of the inside microscopic look of what's going on in the skeletal muscle. And in our next lecture, we'll really get into how this is going to actually work. And we'll walk through these steps.